My name is Danielle Daunhauer. I am a senior biology major at ABAC. For my research, I partnered with the Veterinary Diagnostic Lab here in Tifton. I had the opportunity to work with them on their project concerning otitis externa in canines, which is more often termed as ear infections. Just to give you a brief background, ear infections are among the top 10 ailments seen by veterinarians, and it affects up to 20% of dogs. And these microorganisms that cause these ear infections are usually opportunistic. And they're also zoonotic, which means they will be transferred from your pets to you, which includes resistant strains such as MRSA, which is a very difficult infection to get rid of. So for our objectives, we wanted to determine the most common bacterial and fungal isolates in otitis externa in the United States. We also wanted to analyze the antibiotic susceptibility profiles of these isolates to determine what antibiotics they are susceptible and resistant to better improve the effectiveness of treatments for canines. So for this research, we took a bunch of ear swabs from all over the United States that were submitted to the veterinary laboratory. And once we get the samples in, the lab techs will then take the isolate and plate them using 3 different media plates. We'll have blood agar, McConkey, and PEA. This here is an example of the blood agar plate. And depending on the results from these plates, they will then determine what other series of testing needs to be done to further identify the isolate. And some examples of these further testing are the catalase test and the coagulase test. Here we see the catalase test, which is an enzyme that some bacteria have. The reagent is hydrogen peroxide, and they will drop hydrogen peroxide onto the isolates, and if it bubbles up like this, that is a positive identification for catalase enzyme. Here's our coagulase testing. You can see the liquid here is coagulated, and here's more fluid, so this would be our positive identification for coagulase enzyme. So once the isolate is identified, they will then do AST testing on Mueller-Hinton agar plates, is what you see here. And what they'll do is they will culture these bacteria onto these plates and then stick these little antibiotic discs onto the plate that contains the antibiotic and then measure the zone of inhibition, this little clear area here, and determined by preset standard guidelines, they will measure this diameter and determine whether that isolate is susceptible or resistant to that specific antibiotic. In our results, you see the top 7 bacterial isolates that we found in our study. And here are the graphs of the AST profiles. On the y-axis, you have the percentages. On the x-axis are all of the different antibiotics that were being tested. In green are the sensitive results, and in blue are the resistant results. So in our discussions and conclusions, just a summary of what you see in the graphs. If you're interested in the specific percentages of these susceptibility and resistance. And for further research, we want to finish the AST profiling on all of the different bacterial isolates that we found, and also to analyze the data for the single and co-infections that are causing ear infections in canines.